Shall we start? Yeah. Good afternoon, everyone. I'm Elizabeth Heitman. I'm a co-chair of the study and this committee, and along with my co-chair, Dr. James Collins, committee members, and the staff for the study, I'd like to welcome everybody this afternoon. The task that this group, this committee is undertaking for the National Academies of Sciences, Engineering, and Medicine is to review the state of the science of gene-drive research and provide general principles that will guide responsible practices in gene-drive research. This meeting is an open, on-the-record session. Members of the public, including journalists, are invited to attend as observers. This is, however, not a news conference, and the committee will not be taking questions from the floor. We do have a designated time, approximately 4 p.m., for members of the public to share brief comments with the committee. They're welcome to share comments, ideas, concerns. If you would like to give a brief statement, please, if you have already registered, that's fine. If you have not, please speak to Dr. Keegan Sawyer, the program director, who's in the corner there at the break. And if there's anyone listening online who'd like to make a statement, please send Dr. Sawyer an email with your name and affiliation. That email is ksawyer, K-S-A-W-E-R, @nas.edu. Or you may also email Dr. Lauren Rugani at LRUGANI @nas.edu. For reporters present, if you'd like to talk with members of the committee, we'd like to ask you kindly to meet with us through Dr. Sawyer and talk to her also during the break. We would like to stress that this is an information gathering session. The committee is just starting the process of assembling materials that we will examine, and we will discuss them in the course of making our findings and conclusions and recommendations. This is a process that started only this morning, and as a result, we ask everyone to please be mindful of the fact that we've made no conclusions. We've not even really had any kind of formal discussion about what the agenda will fully be. And it would be a mistake for anyone to leave here thinking that we've come to any conclusions at this point. We may, in fact, ask probing questions at this session of some of the presenters. These questions, however, do not necessarily represent either the opinions of the person asking the question or any kind of position of the committee or the academies. The session is the first of 4 meetings that will involve the committee's deliberation and drafting of the final report. These deliberations meetings will take place between now, July, and March 2016. Our second meeting, to be held October 28th, will include a one-day public workshop at the original Academy's building on Constitution Avenue, which everyone is welcome to attend. Once the draft report is written, it will go through a rigorous external independent review process by experts who are anonymous to the committee. The committee must respond to this review and make appropriate revisions that adequately satisfy the Academy's report review committee before the report will be finalized. The anticipated release date of the report is April 30th, 2016. What we'd like to do now is to have the committee members briefly identify themselves, introduce themselves with their name, their institutional affiliation, and their focus of their work. And we'd like to start with a committee member who's not here except by phone, and that's Dr. Lynn Rediford. Are you on the phone, and can you unmute yourself?
Yeah.
There you are. Hello.
Okay. Hello. Hello. Can you hear me?
Yes, you're great.
Okay. Can you hear me?
Yes, we can hear.
Oh, okay. I'm Lynn Rutherford. Right now I'm at the Howard Hughes Genelia Farm of the Howard Hughes Medical Research. I was professor of biology at the University of Washington for a long time. and I work mainly in basic research on hormones and insect development and a little bit on insect reproduction.
Wonderful. Thank you, Greg. Go around the table from here out.
Hi, I'm Greg Kaepnick. I'm an ethicist training in philosophy at the Hastings Center, which is a nonprofit, nonpartisan research institute. I'm editor of their journal, the Hastings Center Report, and a scholar there, and I've done work on ethics of biotechnology.
Good afternoon. I'm Jason Delborn from North Carolina State University. I'm Associate Professor of Science, Policy, and Society.
Good afternoon. I'm Steve Higgs. I'm an Associate Vice President for Research at Kansas State University, Director of the Biosecurity Research Institute, also Editor-in-Chief of Vector-Borne and Zoonotic Diseases. My background is in research, education, and training on viruses transmitted by mosquitoes.
Hello, I'm Vicki Chandler. I'm Dean of Natural Sciences at Minerva Schools at KGI. My background is fundamental biology and genetics with a focus on plant genetics, working in crop plants.
Hello, I'm Joyce Tate. Excuse me, I'm Joyce Tate. I'm director of the Inogen Institute at the University of Edinburgh, where we work mainly on the interdisciplinary interactions between innovation systems, regulation and governance, and stakeholder perspectives in the development of the life sciences across the board, from GM crops to stem cell therapies.
Good afternoon. I'm Nicole Hache. I'm a research associate professor at the University of Notre Dame. I'm a medical entomologist, and my research program focuses on the evaluation and development of vector control products for control of arthropod-borne diseases of international importance, such as malaria and dengue.
Hi, my name is David Winnikoff. I'm an associate professor of bioethics and society at UC Berkeley. My training is primarily in law, but also bioethics and science and technology studies, where I study the ethical, legal, and social aspects of emerging technologies. I'm currently on leave at Berkeley and working as the secretary of a working party on biotech, nanotech, and converging technologies at the OECD, Organization for Economic Cooperation and Development. Thanks.
My name is Lisa Tannehill. I'm an associate professor in the Department of Animal Sciences at the University of Maryland. I'm a vertebrate developmental biologist, and we use the chick embryo to study early embryonic patterning.
I'm Wayne Landis. I direct the Institute of Environmental Toxicology at Western Washington University in Bellingham, the state of Washington. I do ecological risk assessment.
Good afternoon. My name is Anne Kingiri from African Center for Technology Studies, ACTS, based in Nairobi, Kenya. This is a knowledge think tank which is looking at brokering policy and practice. My area of interest is regulatory science, but I'm trying to look at new innovations right now from an innovation perspective. And I'm also bringing in the inclusivity aspect of innovation in this discussion. Thank you.
And I, again, am Elizabeth Heitman from Vanderbilt University Medical Center, Center for Biomedical Ethics and Society, where I'm primarily an educator and an education researcher related to research integrity and the responsible conduct of research and research ethics.
Thanks, Liz. And I'm Jim Collins, professor in the School of Life Sciences at Arizona State University, where our lab works on the evolutionary ecology of emerging infectious diseases, as well as issues in ecological ethics. Along with Dr. Heitman, I co-chair this committee, and I'm also chair of the Board in Life Sciences for the National Academies. So without further ado, what we'd like to do is start in with a section on perspectives from study sponsors, context, and expectations for this NAS study. I want to remind all the speakers that we have to keep everyone on strict time limits because we do have speakers coming in later remotely. And so for this first cluster of speakers, we have 5 minutes assigned to each. And we're going to start with Carrie Wolenitz from the National Institutes of Health. So if you could come on up here. Because we're videoing this, we're going to ask the speakers to work from here. And then at the end, for questions, we'll bring the speakers up to the front table.
Thank you, Dr. Collins and Dr. Heitman, and to the Academy for giving me the opportunity to speak today. And thank you in advance to all of the committee members for the work that you're about to embark on. We greatly appreciate it. I'm from the National Institutes of Health, and I just wanted to share a little bit of our perspective on where we see this study sort of concentrating its time. We're here, of course, today to talk about gene drives, which is an emerging technology with an incredible amount of promise and potential when it comes to public health, whether it is the transformation of disease-carrying vectors, the modification of wild-type populations, or the applications of gene drives for potential therapeutic production, compounds in plants or other organisms. Certainly, these are not my slides, but they look delightful, so I look forward to seeing them. I'm going slideless today, so forgive me. At the end of the day, NIH's overarching goal with this or any emerging technology, which we spend time thinking about or encourage others to spend time thinking about, is really making sure we're supporting the best science to advance human health. And that we're doing so under the highest ethical standards and approaches. And so we really look at any sort of emerging technology with a real kind of dual use potential through sort of a tri-focus lens, that of the promise and pitfalls of the science, as well as the potential risks and concerns, whether it's in an ethical, legal, security, whatever sphere, and the ability of our existing oversight system to support the science while mitigating whatever risks might be raised. So when it comes to the work before you, what at the end of the day we're hoping to get out of it is sort of under those three general broad strokes. When it comes to the science, we're really hoping that at the end of the day we'll have a solid understanding of the genome editing tools that are really at the heart of gene drives and a better understanding of this emerging technology. Not only the state of the science, but how effective these technologies are when it comes to achieving their aims. What are the potential for unanticipated and undesirable effects when it comes to target or non-target organisms and their ecosystems? What do we know about techniques for limiting the spread of gene drives, both within and between populations? And how do we reverse unintended effects if they occur? What's our backup plan there? An analysis of what the potential applications are. Being the NIH, of course, we're particularly interested in the context of human health, though certainly that's not the limit of this technology. What are really the likelihood for success weighed against the potential for unintended consequences? I think it's clear that any new technology poses uncertainty in terms of impact and risk. I mean, that's part of the world in which we live. So, when it comes to gene drive research, some of the things we're thinking about is when you've got the differentiation between contained research and the biosafety risks involved there, one sort of aspect to look at, and then field research, when the potential to pose sort of broader societal or population ethical issues there, whether it's the impact on human health, questions about site selection and participation in the bioethical issues involved there, potential loss of genetic diversity in wild populations. Let it not be said that NIH cares only about humans. And potential ecosystem or environmental consequences of any such research. And finally, well, not finally, but sticking with that sort of threefold perspective. Yep. Oh, 1 minute left. I'll get through this fast. We're very interested in how this fits within our current policy framework. What's the adequacy of the framework and oversight mechanisms that we currently have? At the end of the day, we're really hoping that the study will provide us and the research community with some guiding principles to really guide responsible practices in gene research. It would be most useful from our perspective if these principles could apply both in laboratory settings as well as all the way through the spectrum of field release, and that you think of all the stakeholders involved, the investigators, the research institutions, the research funders, and the regulators, because all of them clearly have a point of view in this particular sphere. And with that, hopefully I'm under my 1-minute warning limit, and I look forward to the conversation.
Well done. 3 seconds to go. The NIH has set a standard, a very high bar here in terms of presentation. So let's go on now to Stephanie James from the Foundation for the National Institutes for Health.
Good afternoon. I want to echo Carrie's thanks to everyone for being here. I think probably I should make it clear that the Foundation for NIH is a public charity. It is not a government agency. Under the Grand Challenges in Global Health initiative, in collaboration with the Bill and Melinda Gates Foundation, my organization, the Foundation for NIH, has been supporting research since 2003 to develop biological strategies to deplete or incapacitate insects that transmit human diseases. The goal of this research is to create methods for the control and prevention of vector-borne diseases that are safer, more effective, more affordable, and more sustainable than the currently available vector control tools. The original charge given to us in this research effort by our Scientific Advisory Board was that any new strategies to be developed must ensure effectiveness in the field, safety, and social and environmental acceptability. This effort, the Grand Challenges in Global Health, has included research on genetically modified mosquitoes. One such project aims to develop gene drive mechanisms to suppress Anopheles gambiae populations in Africa to levels that are incapable of sustaining malaria transmission. As you know, malaria was responsible for more than 400,000 child deaths in Africa last year. And it is widely acknowledged that additional new tools and control methods are required. Recent mathematical modeling results suggest that the research we're currently supporting has the potential to reduce malaria transmission dramatically and long-lastingly, even in areas of Africa with a strong force of infection and with very little in the way of alternative malaria control infrastructure. So we feel very strongly that gene drive research has exceptional potential for adding to our arsenal of tools to control vector-borne diseases. However, from the beginning of our role in this kind of research, we've taken the charge of ensuring safety and acceptability very seriously and have worked to keep the planning and of the development pathway for this new kind of technology ahead of the science. And that would include the planning for regulatory and stakeholder engagement aspects of the research. For example, our efforts have contributed to publication of the World Health Organization guidance framework for testing genetically modified mosquitoes, which proposes a phased testing pathway analogous to the development pathway for other new public health tools. Also to expert consensus recommendations for the conduct of contained field trials for vector mosquitoes incorporating gene drive systems, as well as for a number of other publicly available papers relating experiences with field site selection, regulatory and community engagement requirements. The accessibility of new and more facile molecular methods has quickly progressed the state of this this research, expanding the capability to create organisms incorporating gene drive to many laboratories worldwide. The possibility of engineering wild populations of many different species creates unprecedented opportunities, not only for the control of vector-borne diseases, but also opportunities in fields like conservation, ecology, and agriculture. However, not all scientists and their institutions engaged in this research may have the training or the resources or the preparation to be aware of the broader array of requirements surrounding this kind of research, or to respond to these requirements at the same deliberate pace that has thus far been pursued. Inadvertent mistakes, as well as impractical or ill-conceived advice on how to avoid those mistakes, run the risk of confusing the field and setting back progress. Progress may also be impeded if we fail to efficiently identify well in advance any regulatory or other oversight gaps and work effectively together to put the necessary mechanisms in place before the scientific need arises. For technologies that will be used primarily in other countries, this requires early and substantial input from those countries. So FNIH and our partner, the Gates Foundation, believe that this is a pivotal point. At this time, there is a need for the kind of thoughtful, unbiased, and thorough approach to examining the needs in gene drive research that we expect from the National Academy of Sciences. Our goals in supporting this study, and this is my slide, are for the panel to review the state of the science of gene drive research that relies on either genome editing technology like CRISPR-Cas9 and other endonucleases, or other genetic modification approaches that have been proposed, and both provide an independent assessment of the promise of this research and also evaluate the suitability of the proposed methods for risk assessment and risk mitigation, including the appropriate balance of caution and practicality. Using appropriate case studies, we hope you will review the existing mechanisms for oversight of research on GMOs, including institutional biosafety and ethics committees, national or local regulatory authorities, and international instruments such as the Cartagena Protocol, as well as the existing guidances for risk assessment of GMOs, and determine whether, if these are properly adhered to, they are sufficient to evaluate the potential environmental, public health, and ethical considerations of individual applications on gene drive technology and safeguard against accidental or intended misuse. If they are not, if you find they are not, then we would hope that you'll be able to help us identify any significant gaps in oversight for handling of organisms containing gene drive at any stage of development. And finally, we hope that you will synthesize general principles to guide responsible practices in gene drive research addressing the development of this technology in the laboratory setting through to field releases, which would be suitable for use by investigators, their institutions, their funders, and by regulators. Thank you.
All right, thank you very much. Let's move on now to Phil Randazzo from the Bill and Melinda Gates Foundation.
So I'd like to thank the committee for inviting me here to speak, and I will just acknowledge our foundation's partners at the FNIH, who we've been closely working with all these years. So an estimated 198 million people suffered from malaria in 2013, 584,000 died, and about 90% of the deaths were in sub-Saharan Africa. 75% of the deaths were among African children under the age 5. So that's more than 430,000 young children dying in sub-Saharan Africa each year. That's about the same number of babies born in Spain each year, and about the same number born in Texas each year. So one of the hardest things I've ever have seen is was a 6-month-old child in a rural clinic in Kenya, lying alone, gasping for breath, and dying of malaria. So the human suffering is real, tragic, and unforgettable. So we can all agree that malaria is a scourge in Africa and globally, taking a tremendous human toll of death and suffering. And we've made huge progress over the past 15 years in identifying and scaling up interventions to reduce the burden of malaria by more than 50%. But access to bed nets, insecticide spraying, timely diagnosis, effective treatments, and a future vaccine may not be enough to end malaria forever. Current tools and treatments are insufficient to achieve elimination in many countries. And the cost of just maintaining the present interventions has reached several $billions a year. So it's unlikely that we'll be able to sustain the political will that supports this high level of investment unless we provide donors with a clear endgame for malaria. So that's why we need to act now to develop strategies that can accelerate the elimination and ultimate eradication of this terrible disease. Malaria has some unique characteristics that require sustained zero-case elimination. In Sri Lanka back in 1953, there were 92,000 cases of malaria. A decade after intensive efforts in Sri Lanka, there were only 17 cases. At that point, malaria programs were no longer aggressively maintained, and there was a rebound effect with 1.5 million cases between 1976 and 1978. So this resurgence is a phenomenon that's occurred in dozens of countries. And because of this phenomenon of resurgence, we began to look for an intervention that was self-sustaining and could be useful in regions where delivery is difficult and where resources are scarce. Thus, we began funding a program back in 2005 that would target a few mosquito species that transmit malaria in a self-sustaining, easy-to-deliver, low-cost manner. There are a total of about 3,400 different mosquito species, and this project is developing gene drive technology to specifically and precisely suppress the population of two major malaria-transmitting mosquito species in Africa, Anopheles gambiae and Anopheles fenestis. Excuse me. Doing so will likely have a substantial impact on malaria transmission in Africa. So over the past 10 years, we've invested over $40 million into this one project. The project first worked with homing endonucleases as a means of creating gene drive in the laboratory. We have substantially funded the stewardship aspects of this program at what we believe is a very high level, with top experts in safety, risk assessment, SOPs, training, containment, regulatory, ethics, and community engagement. And so this has resulted in 10 years of accumulated experience. Recently, CRISPR technology has come into play, and this has changed the game. Before CRISPR technology, gene drive research could only be performed in a few capable, well-funded labs. Now gene drive research can potentially be performed in any lab with molecular biology capabilities. In fact, gene drive research can now be easily performed in a plethora of different organisms. So we have close alignment between the investigators of this program we are funding to assure that this work complies with the current and emerging safeguards appropriate for all stages of the work, and with ongoing engagement and guidance provided by relevant stakeholders. And that has been developed longitudinally over the duration of the program. However, having funded this research over the past 10 years, we feel that it's important that academic labs are aware of and abide by a set of standards in order to safely work in this area. And so we think it's critical to develop an unbiased guidance that will allow potentially transformative, life-saving applications to proceed while ensuring that clear standards are developed to ensure safe and ethical use of such technology. Thank you.
Thank you very much. Let's move on now to Daniel Wattendorf from DARPA, Defense Advanced Research Projects Agency.
Good afternoon. I'm Dan Wattendorf from the Defense Advanced Research Projects Agency. I'm in the Biological Technologies Office, and I thank you for inviting me here to speak today. DARPA is particularly interested in this area because we feel like there are two aspects of this technology that are critically important to both deliberate on the capabilities and the security surrounding them. One is what is exceptional about gene drives, which is the ability for a genetic element, particularly when it's synthetic or designed by an individual versus natural, to propagate and change a population. And the ability to do that could have security ramifications for the United States and for the globe. Secondly, which is, as Phil mentioned, is the facility to perform these types of experiments and create these capabilities faster than ever has been occurred before because of the emergence technology of genome editing techniques. Because of those two reasons, DARPA is looking at this technology as something that is capable for great benefit and is also potentially something that could be a security threat, particularly at ecosystems and population levels for organisms that reproduce quickly, such as agricultural or animal or plant. So I think these circumstances are key for us to be able to understand the deliberation of the community that is looking at the best way to inform the process of how to investigate these capabilities most effectively. However, DARPA has a very specific role in the community to not admire the problem and be able to look at all the potential ramifications, and even for both capability in good and in bad, but also to be used as a tool to create technology that could either mitigate the threat or ensure that its success works in a safe way. So it is certainly within possibility that DARPA would take on efforts to invest in gene drive technologies in ways that would either provide it to be a safe tool or a safe technology, and ways that would be able to mitigate threats when used by our adversaries or by others who are malinformed, or by doing it potentially with malfeasance in mind. So it is very helpful for us to not only understand the deliberations of this community, what you decide to do as you move forward, but also to be able to understand how you could point to what are technologies that you need to do to either make it safer or make it able to us to mitigate the threat. So we are particularly looking at not only the outcome of this group, but potentially even specific recommendations to be able to understand where you see that those technologies being able to be helpful, because we may be required to actually invest in technologies to improve this capability during the process of elaborating the challenges and benefits of it. Because we may not have the time in this case to actually wait for and make calls for certain scientific actions in communities to deliberate. We actually may need to be working on technology solutions right now. And the alacrity of our institution to be able to do that is at hand. And that's why we are here today, to be able to follow that. Thank you.